Okay, so welcome back from the break. Let's take a look at the concept check questions and then we'll move on. So if both parents have to pass on a faulty version of the allele in order for a child to have the condition, that has to be a recessive trait, right? Because we know that means the individual that has the trait is going to be homozygous recessive under Mendel's principles. And remember, Garrett is simply applying Mendel's principles to his condition. And he actually said this in 1902. So you can see that he was way, way ahead of his time in terms of his understanding of inherited conditions and what they represented. What would a polypeptide product of a 30 nucleotide polygene messenger RNA be? Well, let's go back to the genetic code very quickly. And let's find GGG. So we start on this side. So there's the first G. That's the first G of the codon. Here's the second G. And then we're going to come down here and find the third G. And there it is there. GGG. And that's for the amino acid glycine. So if it is a 30 nucleotide messenger RNA, and they're all Gs, that means we've got 10 three-letter GGG codons. So we're going to have a 10 amino acid polypeptide, and it's going to be a 10 glycine polypeptide, because GGG is the codon for glycine. Okay, so let's move on. So the first thing that we're going to look at is transcription. And so when we start thinking in terms of transcription, we are making a polymer. In this case, we're making an RNA. So the enzyme that is involved is called RNA polymerase. And RNA polymerase does all of the work here. So there are no other enzymes to assist it in the process of transcription. So RNA polymerase, once it attaches to the DNA, opens up the DNA by breaking the hydrogen bonds between the adjacent nucleotides. But it's not trying to open up the whole of the DNA molecule. It's only opening up a local area of about 20 to 30 nucleotides long. And it doesn't need a primer. It can simply start assembling the transcript using RNA nucleotides without requiring a primer. So there are some differences here. It's only going to copy the template strand. It does its own heavy lifting in terms of unwinding and unzipping the DNA, but it does so only locally in a region of around 20 to 30 nucleotides long at any given point in time. And it doesn't need the DNA to be primed in order to be able to start creating the transcript. And of course, being RNA, as the note indicates, U's are going to be incorporated in where we would normally expect a T in the DNA as we make the complement. So there are three stages that are recognized: initiation, elongation, and termination. When we talk about translation, you're going to see the same three stages. So it's really important now that you start to differentiate between these three stages as they occur in transcription versus these three stages as they occur in translation. The names are the same, but clearly what occurs in each of them is going to be very, very different. So in initiation, the basic process involves the binding of the RNA polymerase to a region that we describe as being upstream from the gene. So we need to identify what we mean by upstream and downstream. So remember that the DNA is always read 3' to 5'. So make sure the pen is working. DNA is always read 3' to 5'. And that is said to be downstream. So if you're going 5' to 3', that would be upstream. So if we are reading the template here, 3' to 5', which is this strand here, this is the template strand, then going in the other direction, this is 3' to 5', going in the other direction would be going 5' to 3'. So the location where the RNA polymerase binds must be upstream of the gene unit or the gene sequence. So the RNA polymerase binds upstream of the gene unit, opens up the DNA, and starts reading the gene sequence from the initiation point of the gene, and starts assembling the transcript on the basis of the sequence in the template strand. Elongation is the continuous process of the RNA polymerase working its way down the DNA, sequentially opening up the DNA by unwinding and unzipping and assembling the transcript. As the RNA polymerase continues on down, creating the transcript, as the DNA exits from the RNA polymerase, it re-anneals, meaning the two strands connect together again. That's called re-annealing, and then rewinds again, because we don't need to keep it open. We're not trying to copy both strands, we're just copying one strand, and the RNA polymerase is doing that. Termination occurs particularly in bacteria when the RNA polymerase encounters a sequence of nucleotides at the end of the gene unit called the termination sequence that triggers the ending of transcription and the disengagement of the RNA polymerase and the release of the transcript, which in the case of the bacteria is the intact messenger RNA. In the case of eukaryotes, there may be a terminating sequence. It may be a little bit further downstream from the end of the gene, and it usually is located a little bit further downstream from the gene. Or there may not be a termination sequence immediately adjacent to the gene unit. It may be some distance from the gene unit. But at some point, the RNA polymerase will cease transcription and will disengage from the DNA and release the transcript. That's not a big deal, because remember, in eukaryotes, subsequent to transcription, we're going to be processing the RNA molecule anyway to create the final messenger RNA. So having a termination point that is downstream from the gene and maybe at some distance from the gene is not a big deal, because we're going to be processing the RNA, the transcript anyway. So this is the general idea of what goes on. In terms of a little bit more specificity, we do recognize that the region on the DNA that the RNA polymerases bind to is referred to as a promoter. Now, the nature of the promoter in prokaryotes versus eukaryotes is a little bit different. So in bacteria, genes are organized into units called operons. So in bacteria, genes are organized into units on the basis of what those proteins are involved in. So the first of these organized units, these operons to be recognized, is something called the lac operon. Lac referring to the metabolism of the sugar lactose. So there are three proteins that are involved in the metabolism of lactose. The bacteria only needs those proteins when lactose is present. So having these genes all controlled by a single promoter means that you only produce those proteins when lactose is present, when you require them, and you produce all three of them together as a consequence of being controlled by a single promoter. Now, between the promoter and the beginnings of the genes, so I'm going to move this over here. Oops. So just to explain this note, not only do you have a DNA sequence called the promoter that the RNA polymerase is combined to, but between the promoter and the actual beginning of the sequence of genes is another region called the operator. And there are repressor proteins that bind to the operator and block the ability of the RNA polymerase to carry out transcription. So if the repressor protein is bound to the operator, the gene is functionally turned off. And so the RNA polymerases in bacteria can bind to promoters without any assistance. But if the operator is blocked by the repressor protein, it doesn't matter that the RNA polymerase may bind to the promoter. It cannot move past the operator because of the presence of the repressor protein, and therefore the gene is turned off. However, if those genes are required, the repressor protein is inactivated. And once it's inactivated, it no longer binds to the operator. So when the RNA polymerase binds to the promoter, it now has free access to the gene. It can move down the operator, down the promoter, past the operator and on into the gene to start transcription. So the gene is effectively turned on. And it's all about how these repressor proteins are regulated as to how the operons are turned on or turned off. So in the case of the lac operon, the repressor protein is active in the absence of lactose. So long as there's no lactose around, the repressor protein binds to the operator and keeps the gene turned off. In the presence of lactose, the repressor protein is inactivated. The lactose actually binds to it and alters its shape so it can't bind to the promoter. That releases the lac operon to be transcribed because there's now lactose there and the genes are required. And that's the typical way in which the operons are controlled in a bacterium. But there are other operons that only become active in the absence of a molecule. So in the case of amino acids, for instance, and the first example of an operon that's involved in this is what is known as the TRP operon that produces tryptophan. The enzymes there actually make the enzyme the amino acid tryptophan. So in the presence of tryptophan, the repressor protein is active and blocks the operon. Because if you've got tryptophan, you don't need to make it. But when tryptophan is absent, the repressor protein becomes inactive and the operon is active and transcribed. So that's the bacterial system in terms of initiating transcription and controlling the initiation of transcription. In the eukaryote system, which is what is diagrammed in the textbook in detail. And by the way, let me just add in here, bacteria only have one RNA polymerase. That does all the transcription. Eukaryotes, on the other hand, have 3. So RNA polymerase 1 and 3 are involved in transcribing genes that result in RNA molecules but don't lead to polypeptides. RNA polymerase 2, as you'll see in the diagram, is the one that transcribes genes that are going to lead to the production of polypeptides. So let's take a look at that. So in the promoter region of a eukaryotic gene, on the non-template strand, so on the non-coding strand, in the promoter region, there's this repeating sequence of nucleotides, TATA. And this has become known as the TATA box, TATA, TATA. And this is important because a protein called a transcription factor will bind to this. Now, the diagram in the book does not go into as much detail as I think you ought to know about the switching on of the genes in a eukaryote, because if we look at the DNA sequence, here's our gene, here's our promoter region. Somewhere down here in the DNA, there are a pair of, I think I'll change the color on this. Oops. Take that out. There are two DNA sequences down here called enhancer sequences. And you can see that these are at a distance from the actual promoter region. So the way this is dealt with is for the DNA itself to actually bend around. So I'm going to erase this a little bit and show you how this works. So the DNA bends around so that the enhancer sequences are now near the promoter region, and there's actually a protein that binds in here called the DNA bending protein. It actually binds to the DNA and bends it around to bring the enhancer sequences adjacent to the promoter region. That then allows transcription factors to bind to this, to then bind to the TATA box and adjacent to the TATA box, which then allows the DNA polymerase to bind to the promoter. So it's a little bit more complicated, but that's because the level of control over transcription in eukaryotes is substantially more complicated. And the reason why is because many genes in eukaryotes function as families, but the genes are not located near each other or organized into operons as they are in bacteria. So the ability to be able to turn on multiple genes all at once depends on the transcription factors that are involved and the ones that bind to particular enhancer sequences and therefore activate particular genes. And different combinations of transcription factors control different families of genes. And one of the reasons why different types of animals show different levels of complexity, yet seem to contain similar numbers of genes. So we can compare ourselves to fruit flies. Fruit flies contain somewhere around 15 to 17,000 genes. Humans contain no more than 21,000 genes. That's the current estimate. So there's not a significant difference in terms of the number of genes in a fruit fly versus a human. Maybe a couple thousand genes. But where the big difference lies is just how much we can do with those genes. So there are a set of genes called master control genes. The proper name for them is HOX genes, H-O-X. And these are the genes that control other genes as master control genes. Fruit flies only have one set of those genes. Humans and animals that we would describe as vertebrates, so anything with a vertebral column, have 4 sets of those master control genes. So you can imagine that having one set of master control genes versus 4 sets of master control genes determines how much you can do with a basic set of genes. If you've only got one set of master control genes, there's not much you can do with them compared to an organism that has 4 sets of master control genes. There's so much more that you can do with them. So the reason why humans are so complex is not because we have a huge number of genes, it's because we can do so much more with the genes that we have because of these master control genes. And those master control genes, they are the transcription factors that are initiating the binding of the RNA polymerase to the promoter. So it's the combinations of transcription factors that determine which genes get activated and allow the RNA polymerase to bind to begin the process of transcription. So initiation is far more complicated in eukaryotic organisms as a consequence of this. In terms of elongation, it's pretty much the same whether we're talking about bacteria or eukaryotes. It's all about the RNA polymerase opening up this transcription bubble, if you want, this local region where it's unwound and unzipped the DNA, and it's creating the transcript using the template strand as the template for building it, adding nucleotides to the 3' end of the growing transcript. And it continues to slide along, unzipping the DNA and unwinding it. And as the DNA exits from the RNA polymerase again, it reanneals, hooks back up together again, or rezips, and then rewinds. And then, as I said, termination, it depends. In the case of bacteria, there is always a terminator sequence at the end of the gene sequence that triggers the end of transcription and the release of the transcript, which, remember, is now a functional messenger RNA and can be translated immediately without further processing. In the case of eukaryotes, there is an additional piece of RNA that's added to the tail of the gene. It's called a poly-A tail. And the length of that is determined by a sequence within the gene itself. We'll talk a little bit more about that when we do the RNA processing in a few moments. But somewhere further down from the end of the gene, you're going to find a termination sequence that will eventually trigger the end of transcription, but not until the poly-A tail has been added in as a consequence of the nucleotide sequence contained within the gene, the body of the gene itself. So again, the eukaryotic process, a little bit more complicated than the bacterial process. So let's stop again for a moment, because that's translation, fairly straightforward process. And look at these two concept check questions. So what is a promoter? Is it located upstream or downstream from the transcription unit? And how does the initiation of transcription differ between prokaryotes and eukaryotes? So remember, the promoter is the region of the DNA that the RNA polymerase actually binds to, and it is always located upstream from the gene unit or the transcription unit. So what about the initiation of transcription between prokaryotes and eukaryotes? Remember, in prokaryotes, the RNA polymerase does not need assistance to bind to the DNA. It simply binds to the promoter. So how do we stop transcription or start transcription? Through the operator sequence that lies between the promoter and the gene sequence, or the operon sequence. So the operator can either be blocked by a repressor protein or not blocked by a repressor protein. If the RNA polymerase binds to the promoter and there's a repressor protein on the operator sequence, then it is blocked and the gene is turned off. If the if the repressor is inactive and not bound to the operator, the RNA polymerase is then free to carry out transcription. In eukaryotes, remember, we require transcription factors. The enhancer sequences located elsewhere on the DNA get bent over by the bending protein to come adjacent to the promoter region. The initial activator proteins bind to that, which then triggers a transcription factor to bind to the TATA box, that TATA repeat on the non-template strand or the non-coding strand within the promoter. That in turn attracts other transcription factors that also bind to form a complex that attracts the RNA polymerase, and in this case it's RNA polymerase 2, that binds to the promoter and begins the process of transcription. Very different processes, but something that you need to be aware of. So let's move on now and take a look at translation. Oh, sorry. For translation, we've got to look at the editing of the RNA, the post-translational editing of the RNA. So why do we need to edit the eukaryotic transcript post-translation? Well, for two specific reasons. Number one, it turns out that the cytoplasm is a very, very hostile environment for the messenger RNA. So we need to protect the messenger RNA against the enzymes in the cytoplasm whose goal it is to chew up the messenger RNA. So there are specific steps that are taken to protect the messenger RNA. The second reason, so let me change the color on this, back to red. So we need to protect the mRNA from being degraded in the cytoplasm. So that's the first reason. And the second reason is we need to remove non-coding sequences which are called introns. And then once we've taken out the introns, we need to splice together the coding regions, which are called exons, to form the final messenger RNA. So there is an actual requirement for processing the transcript. This is not something that happens arbitrarily. There are very specific reasons why this needs to take place. So part of the protective mechanism is incorporated during the process of transcription. So it's added by the RNA polymerase. So the poly A tail is of varying lengths depending on the messenger RNA. The actual length of the poly A tail is determined by a sequence of nucleotides in a region of the gene and the messenger RNA itself, because it's actually retained in the messenger RNA, called the 3' UTR, and UTR stands for untranslated region. So it's a section of the final messenger RNA that specifically does not get translated, but it's there. And there are a sequence of nucleotides in that region that indicate to the RNA polymerase how many A's to add to the 3' end of the messenger RNA to create the poly A tail. The length of that poly A tail determines how long the messenger RNA is going to remain stable in the cytoplasm. The 5' cap, I'm going to bring a line down here, consists of a modified G nucleotide. So you may recall that when nucleotides get added to a growing nucleotide strand, 2 of the 3 phosphate groups are removed, and the remaining phosphate acts as the bridge between the nucleotide and the adjacent nucleotide to form the sugar phosphate backbone of the polynucleotide. In this case, the 3 phosphate groups remain on the G nucleotide. That's why we say it's a modified G nucleotide. What does this do? Well, it allows export of the mRNA, protects from degradation, I guess I'll have to put it here, and identifies 5' end to the ribosomes. So the ribosomes need to know which is the 5' end because that's the end that they're going to start translation from. So adding the cap also serves to help the ribosomes identify which is the 5' end of the messenger RNA so that they will then bind to that end and then be able to identify the location of the start codon, which is also an important step. So the addition of the 5' cap, this modified G nucleotide, is really central to the success of translation, because in the absence of that cap, number one, it is unlikely that the messenger RNA is going to be exported from the nucleus. The presence of 5' cap indicates that it's ready to be exported. If it does manage to leave the nucleus, then it's immediately open to degradation because the 5' cap stops the enzymes that would normally chew up a messenger RNA from immediately going ahead and doing so. In the absence of that cap, those enzymes are going to start chewing it up. So you'd lose the messenger RNA immediately. And even if it didn't get chewed up, the ribosomes would struggle to identify which is the 5' end and which is the 3' end, which means they may not assemble correctly on the messenger RNA if they assemble at all, which is going to totally screw up translation. So the presence of the 5' cap is central. So you can see here the regions that I was referring to. So here's the 3' UTR that tells how long to make the poly A tail. So this is what the RNA polymerase is going to read to determine how many A's to add on here. And then at the 5' end, not only do you put the cap in, but you've got this small region called the 5' UTR, again, untranslated region. But this serves to identify where the start codon is, because the start codon, the AUG that's adjacent to the 5' UTR, the ribosomes identify as the start codon. They actually lock on to the 5' UTR and orientate themselves to the AUG adjacent to the 5' UTR. So this and the 3' UTR. So that sequence in the 3' UTR simply identifies for the RNA polymerase how long to make the poly A tail. So you can see that these are really important. So that's the capping and tailing. What about the editing? Well, that's done by a complex of proteins and RNA molecules called spliceosomes. They identify the non-coding regions, the introns, and cut them out and then splice together the remaining coding regions, the exons, to create the final complete messenger RNA. Remember, the capping and tailing has already taken place, so the cutting and splicing is the last remaining step. So this is what the spliceosomes do. They identify where the exon finishes and the intron begins and bind to that region, then identify where the intron ends and the next exon begins, attach to that and cut out the exon, sorry, the intron, and then carry out the splicing together of the exons, having removed the intron. And all eukaryotic genes have some introns in them, some more than others. And you might ask, why do we need introns in them? Well, there's a really good reason for having introns in the genes. But before we get to that, one of the things that we have discovered, and this is the first instance where you will have encountered this, is that not all enzymes are proteins. And this is where we find this for the first time. And we're going to see it one more time before we're done today. These RNA molecules in the spliceosomes are in fact enzymes. They are RNA molecules that act as enzymes, and they are known as ribozymes. You can see the name here, ribozyme, an RNA molecule that functions as an enzyme. And it is these enzymes that do the cutting and splicing, these little RNA molecules that do the cutting and splicing. And so the proteins themselves are actually there just to support the RNA molecules. So it's a different relationship here. Normally we would think that the proteins are doing the work and the RNA molecules are there to support. No, in this case, the RNA molecules are doing the work and the proteins are there to act as the support. And in fact, we now recognize that there are a whole host of little RNA molecules that are involved in gene regulation. We don't get into the details in this class, but you may get involved in some of that in some more advanced biology classes if you take some, or you may talk about these in one of the other classes that you'll need to take before you get into the health sciences. It depends on your professor as to whether they'll talk about that. But if you get into more advanced biology classes, you're getting a biology degree or a microbiology degree or something like that, because you're planning to go to medical school, then yes, you will talk about these tiny little RNA molecules that are involved in gene regulation. We actually think that in terms of regulating cellular activity, the RNA molecules may turn out to be far more important than proteins in terms of regulating cellular activity, because they're regulating cellular activity at the most fundamental level in terms of regulating transcription and to some degree translation, but very much they're involved in regulating transcription and gene activity. And so by regulating gene activity, they can very definitely regulate all sorts of cellular activities because of the knock-on effect. So this is the first time that you've encountered an RNA molecule functioning as an enzyme in the spliceosomes. So why have these introns? What's the value of the introns? Well, the answer is in this diagram. So you can see here that in terms of the polypeptide that's being generated here, each of these exons is associated with what is referred to as a domain, a functional region within a polypeptide. So polypeptides and proteins will contain multiple domains, and each of these domains is generally considered to be the product of a single exon. Maybe the product of multiple exons, but generally a domain is the product of a single exon. And you can see that in the case of this particular polypeptide, these exons, exon 1, 2, and 3, when linked together, assemble the domains in the order 1, 2, and 3. But there's no rule that says that this necessarily has to take place. There's no rule that says exon 1 must be followed by exon 2, must be followed by exon 3 in sequence in the messenger RNA. You could, in fact, put 1, 3, 2, or 2, 1, 3, or 3, 1, 2. Any possible combination of exons actually is legitimate. And remember, as soon as you start linking these exons together in different orders, in different combinations, you're creating different polypeptides. And this is why I said back at the beginning, this idea of one gene, one polypeptide, we may have to rethink that a whole lot more because of what we now know about the role of exons and the splicing of exons together to create the messenger RNA and therefore to create polypeptides. And not only is there no rule that says that exons have to be linked together in a particular order, there's no rule that says that all the exons actually have to be linked together anyway. You could drop one out. and just not use it, which opens up a whole other range of possibilities. So what's the big deal out of this? Why is this so important? Well, it's totally relevant to where we find ourselves today. Because one of the things that is so remarkable about our immune system is its ability to recognize so many pathogens. Somewhere around 3,000 to 5,000 individual pathogens can be recognized by the immune system via antibodies. And there are two components to an antibody, what's called the light chain and the heavy chain. And within the light chain is a region called the variable region. And the heavy chain and the light chain of the antibody are the product of different genes. And it turns out that in the variable region of the light chain, within the genes that produce that, there are lots and lots of introns and exons. In fact, the total number of genes involved in the immune system is substantially less than the number of antibodies that can be made. Because for each pathogen that the immune system recognizes, it makes a specific antibody. Just let that settle for a moment. There are not enough genes to account for the number of antibodies that the immune system remembers how to make, and it truly does remember how to make them. So how can it accomplish making all of those different types of antibodies and go ahead and figure out how to make new ones every time it sees a new pathogen? If you've been vaccinated against the COVID virus, what you were doing was exposing your immune system to one of the spike proteins from the virus so that it could figure out how to make a new antibody, an antibody that would work against that protein. And you're relying on the ability of the immune system's genes to come up with a new antibody that works. How does it do that? By shuffling the exons in these genes around. In fact, several research groups have investigated the number of genes that are present and done the mathematics to figure out how many possible combinations of exons can be generated. Excuse me, can be generated. Oh boy. Yeah, let's see if I can struggle through this. Hopefully you can still understand me. But they've done the math, the calculations, to try and figure out how many possible combinations of exons can be generated from the limited number of genes that are present that are involved in making these variable regions. And it turns out that, in fact, the number of pathogens that our immune system can recognize is still less than the possible number of combinations. Not by much, but sufficient so that there is headroom in the number of variability of variable or varieties of antibodies that can be made to allow the immune system to make antibodies against new pathogens as it encounters them. So in fact, the presence of these introns and exons is really important in allowing the immune system to do what it does. It wouldn't work anywhere near as effectively if it wasn't for the ability to shuffle exons around. The other reason why these may be valuable is, again, explained by one of the reasons why there is so much DNA in our chromosomes, yet so few genes. It turns out that in terms of our genome, only about 3% of our total genome represents the 21,000 genes that we know exist in the human genome. The other 97% for a long time was referred to as junk. Well, we now know that those regulatory sequences, the enhancers that I spoke about earlier, and other regulatory sequences are located within that non-gene containing regions, We know that there are remnants of viruses that have infected in the past, probably before humans as a species existed. Fortunately, those virus sequences are crippled. But having all of that DNA simply increases the likelihood of crossing over and increases the frequency of crossing over. And by having exons and introns within a gene sequence increases the likelihood of variability by crossing over. So that would be another good reason for having the introns and the exons. Not only the ability to shuffle the exons around to create a variety of messenger RNAs from a single gene, but also to allow for the exchange of exons between non-sister chromatids during crossing over to create a new variety of genes that allows for even more shuffling. And that seems to be one of the reasons why sexual reproduction exists amongst most organisms, to create protective mechanisms, in the case of mammals at least, and most animals that have immune systems, to create variety in the immune systems to allow them to resist parasites and pathogens, because those parasites and pathogens can evolve at a very frightening rate compared to how slowly we evolve compared to them. So one way to protect ourselves is to be able to create a lot of variation in the immune system. And there's a lot more that I could talk about there, but we don't have time for that. But there's a lot of interesting information coming out and has come out over the last 10 or 15 years about how variation in the immune system plays out. But let's move on. So now that we have edited the messenger RNA and it's ready for export from the nucleus, we are ready to talk about translation. But before we go, let's take another short break and take a look at some concept check questions. And here I am actually going to break again, and I'll meet you on the other side of the break with the answers to these questions. So, given that there are no more than about 21,000, that should be 21,000 genes in a human genome, how is it possible for human cells to make about 75 to 100,000 different proteins? The answer for that should be pretty obvious. If a TV show is an analogy to a eukaryote transcript, what would the introns be? Hopefully you can think about that. You can come up with an answer for that one. What effect would removing the GCAP have on a messenger RNA? We talked about what it's there for. So what would happen if it wasn't there? Let's take a break. I'll meet you back here on the other side of the break for the answers.